Hello, everyone, and welcome to today's session. And today we'll be discussing the theory related to the chapter Cell, the Unit of Life. Okay, so today in this session, we'll be discussing the NCERT section 8.5, okay, 0.4, 8.5.5, and 8.5.6. Okay, and this is the part 5 of the lecture. So before starting this lecture, I just want to give you one information that the images, text and other materials used in the presentations are for education purposes only. We respect the intellectual property rights of the original creators and owners. If you are the owner and have any concerns or issues, please contact us at sathee@itk.ac.in and we shall remove the content immediately. Please note that parts of the content may have been generated using generative AI, which is experimental. So what are the objectives of today's session? So today we'll be talking about mitochondria. Okay, first of all, we'll be talking about mitochondria. So we will understand the structure of the mitochondria and what is the role of mitochondria, what are the functions of mitochondria in cellular respiration. So cellular respiration is the respiration which is occurring within the cell. Okay, it is the aerobic respiration within the cell by which energy is produced in the form of ATP. We'll also talk about the plastids and the type of plastids. Okay, what are the type of plastids? So chloroplast, chromoplast, leucoplast. These are the type of plastids. We'll talk about these plastids and what are their functions. We'll also talk about the ribosome and the importance of ribosome in protein synthesis. We'll talk about the structure of ribosomes as well. Okay, we'll talk about the location of these organelles and what are the function of these organelles in plant and animal cells. Okay, we'll understand the role of these organelles in energy production as well. Clear? So today we'll be talking about mitochondria, which is the NCERT section 8.5.4. We'll talk about the plastid, which is NCERT section 8.5.5. We'll be talking about the ribosomes as well and the function of the ribosome, importance of the ribosome. Then we will summarize this entire lecture. We'll discuss some NCERT based questions related to these topics. So these questions are based on some concepts. We will understand these concepts by discussing these NCERT based questions. Okay, then there'll be some keynotes and interesting facts for you. In the end, there'll be some interesting questions for you. Okay, so you have to think about these questions, and these questions will enhance your critical thinking ability. Okay, so let's talk about the visibility and distribution of the mitochondria. Okay, so how can you observe mitochondria? So just by viewing under the microscope, the mitochondria are not that easily visible. So you have to stain the mitochondria with certain specific dyes. Okay, so mitochondria are not easily visible without staining it with specific stains. A stain which is used to, you can say, a dye which is used to stain this mitochondria is Janus green B. Okay, now the number of mitochondria varies per cell based on the physiological activity of the cell. Means if the cell is active, it will consist of more number of mitochondria. If the cell is not that much active, then it will contain less number of mitochondria. So based on the physiological activity. the number of mitochondria varies per cell. What are the examples? Liver cells, they have 1000 to 2000 mitochondria. So liver cells are quite active. Okay, they have 1000 to 2000 mitochondria. In muscle cells, up to 20% of the cell volume consists of mitochondria. So see, muscles, they require energy to carry out the work. Okay, whenever we are moving, whenever we are talking, whenever we are doing any activity, this activity requires energy. So we know mitochondria produces energy in the form of ATP. Clear? So that's why muscle cells consist of large number of mitochondria. Up to 20% of the cell volume consist of mitochondria. So muscle cells are also known as muscle fibers actually. In case of muscle, they are known as muscle fibers. Then egg cell, it consists of 1 lakh plus mitochondria or 100,000 mitochondria. So these are all very active cells. Now, distribution depends on cellular energy requirements. Clear? So if the cell needs more energy, then it will consist of more mitochondria. Okay? Clear? Like muscle cells, they require more energy to do the work. So more mitochondria will be present in the muscle cells. Okay, let's talk about the physical characteristics of the mitochondria. Okay, physical characteristic means the morphology, the structure of the mitochondria. So how mitochondria looks like? So what is the shape of mitochondria? So mitochondria is sausage-like or cylindrical in shape. Okay, we will see the sausage-like shape of mitochondria. I'll show you the figure as well. Then let's talk about the diameter. So diameter is 0.2 to 1 micrometer. Okay, micro means 10 raised to the minus 6. Okay, what is the average diameter? 0.5 micrometer. Now what is the length? 1 to 4.1 micrometer. So mitochondria, you can see, shows considerable variability in their shape as well as size. So this is the sausage-like shape of the mitochondria. This is the longitudinal section. Okay, so now the various parts of the mitochondria have been labeled as well. So mitochondria is surrounded by two membranes, outer membrane and an inner membrane. Now between this outer membrane and inner membrane, there is a space known as intermembrane space. Then this inner membrane has certain foldings which are known as cristae. Okay. Now you can see the matrix here, which is the fluid present within the mitochondria. So we will talk about all of these terms one by one. Okay, let's talk about the structure of the mitochondria. So mitochondria consists of two membranes, outer membrane and inner membrane. Outer membrane forms the external boundary of the mitochondria. Okay, it is relatively smooth. Okay, they are writing relatively smooth. Whenever you write relative means you are comparing it with something. So you are comparing the outer membrane with the inner membrane. So as compared to inner membrane, the outer membrane is relatively smooth. Okay. Now, outer membrane consists of proteins for molecular transport. So it is helping in the transport of the molecule. So it consists of proteins that help in the molecular transport. Now let's talk about the inner membrane. So inner membrane forms numerous folds known as the cristae. Okay, so what is the function of this cristae? So this cristae increases the surface area for enhanced metabolic activity. Now, what do you mean by metabolic activity? So metabolic activities means biochemical reactions that are going on. So this increases surface area for enhanced metabolic activity. Then inner membrane is very important because it contains important respiratory chain enzymes. Okay, means the enzymes which are involved in respiration, they are present here. Okay, the various complexes are present here. Okay, so when you will study respiration, you will study about the electron transport chain. Okay, and the other enzymes like ATP synthase. So these enzymes, they are present in this inner membrane. These complexes which are involved in electron transport chain, these are present in this inner membrane. Okay, especially in this cristae, which enhance the surface area for the metabolic activity. Okay, so this is the structure of the mitochondria. You can see this is the outer membrane, this is the inner membrane, and these are the cristae. These are the you can say infoldings of the inner membrane. Okay, this cristae consists of the enzymes, and the complexes are present here, various complexes which are involved in respiration. Now let's talk about the internal compartments of the mitochondria. So the inner membrane divides the lumen of the mitochondria into two aqueous compartments. Aqueous compartments means liquid compartments. These compartments are filled with some liquid, some fluid. Okay, now imagine the inner membrane of the mitochondria. Towards the outer side of the inner membrane is present the outer compartment. Towards the inner side of the inner membrane is present the inner compartment. So outer compartment will consist of the space between the outer and the inner membrane. Okay, which is known as the intermembrane space. So what is outer compartment? Outer compartment is basically the space between the inner membrane and the outer membrane. Intermembrane space. Then you have the inner compartment. We'll talk about inner compartment also. Okay. So outer compartment is the space between the outer and the inner mitochondrial membranes. What about the inner compartment? Inner compartment is the innermost space enclosed by the inner membrane. Means the space, okay, enclosed by the inner membrane. The space below the inner membrane. Okay, inner compartment basically consists of the matrix, which is the fluid present within the mitochondria. So this matrix is a dense homogeneous substance. Now it contains multiple components such as it consists of circular DNA molecule. See, mitochondria has its own DNA, and that too circular DNA molecule, not linear DNA molecule. In eukaryotes, linear DNA molecule is present. Here circular DNA molecule is present. So this is a characteristic feature of which organism? Circular DNA molecule. Okay, we'll talk about this. It consists of RNA molecules also. It consists of 70S ribosomes. Okay, so all these are characteristic feature of prokaryotes. Bacteria is a prokaryote. Okay, so you can see here this mitochondria have the features of a prokaryotic cell. So we can say sometime, okay, billions of years ago, maybe mitochondria was a bacteria that entered into the eukaryotic cell. And there was a symbiotic association between this bacteria and the eukaryotic cell. Now, what do you mean by symbiotic association? So, in symbiotic association, both the interacting partners, they are benefited. So, this bacteria stayed within this eukaryotic cell because it was getting benefit, and slowly this became the part of that, this bacteria became the part of that eukaryotic cell, because it was now dependent on this eukaryotic cell. It was gaining some advantage from this eukaryotic cell, so it stayed there, and slowly it lost the other features and it became integrated into the eukaryotic cell. Okay, but the eukaryotes, they have 80s ribosome. Mitochondria have 70s ribosomes. Okay, it has circular DNA molecule. So this, okay, on this DNA and this bacteria, which was this bacteria? Okay, this was a prokaryote. So this is known as endosymbiotic theory. You should read about endosymbiotic theory. This will explain you origin of mitochondria as well as origin of chloroplast as well. Okay. Now this matrix also have protein synthesis machinery as well. So mitochondria can synthesize some proteins as well. Okay. So mitochondria, you can see, consist of their own DNA. So how many types of DNA are there? First, you have the nuclear DNA. DNA which is present within the nucleus, also known as the chromosomal DNA. Then you have extranuclear DNA, okay, which is the mitochondrial DNA. Okay. Then third one is chloroplast DNA. Chloroplast also consists of its own DNA. So mitochondria have its own DNA, it have ribosomes also, and it has other components which are necessary for protein synthesis as well. So mitochondria can produce certain proteins which are essential for mitochondrial functioning. And these proteins are also involved in the electron transport chain and oxidative phosphorylation through which ATP is being produced. Clear? So you can see mitochondria consists of its own protein synthesis machinery. The protein which it produces are involved in electron transport chain and oxidative phosphorylation. So you will read about electron transport chain and oxidative phosphorylation in the chapter respiration in plants. Okay, this is the image of the mitochondria. Now, what are the functions of the mitochondria? It is the primary site of aerobic respiration. So where does respiration takes place? We were talking about the cellular respiration. Cellular respiration means respiration which is occurring within the cell. Where within the cell? Inside the mitochondria. Okay, so all these organelles perform some specific function. So it is the primary site for aerobic respiration. Aerobic respiration means respiration in the presence of oxygen. And what happens during aerobic respiration? Glucose. Okay, this is a respiratory substrate. This glucose is completely broken down to release carbon dioxide, and energy is also released in the form of ATP. Okay, first glucose is broken down into pyruvate through glycolysis, and then pyruvate enters into the Krebs cycle through which the energy is generated in the form of ATP. And this ATP is broken down to release energy, and this energy is then used to carry out all the activities like moving. Okay, so it is primary site of aerobic respiration. It consists of all the enzymes which are involved in respiration, And these enzymes and these proteins, they are present in the inner membrane of the mitochondria. It is involved in ATP production. That's why it is called powerhouse of the cell. Okay, so question can come here that which organelles is the powerhouse of the cell? So answer will be mitochondria. Mitochondria is the powerhouse of the cell. It is involved in protein synthesis. So this we have discussed. Mitochondria consist of their ribosomes. They have their own DNA. They can make proteins. These proteins are involved in electron transport chain and oxidative phosphorylation. Then it is involved in cellular energy metabolism as well. It is producing energy. Clear? Now let's talk about the division in the mitochondria. How mitochondria reproduces, you can say. So again, this is a kind of feature of a prokaryotic cell. Like prokaryotes, like bacteria, they divide by binary fission. In binary fission, what happens? One cell divides into two. So from a parent, two daughter cells are formed. So mitochondria divide by fission. So you can see this is very similar to bacterial cell division. So we have a lot of proof that mitochondria was once a free living prokaryotic cell or prokaryotic organism. Clear? Or which was engulfed by the eukaryotic cell. Okay, so this mitochondria can replicate their own DNA as well. So it is a semi-autonomous organelle. It can produce some proteins by itself, and some of the proteins it requires from outside, which are produced by the nuclear DNA. So it is a semi-autonomous organelle. It is not completely autonomous, it is semi-autonomous. So it can produce some of the proteins by itself. Okay, so mitochondria, as you know, it has its own DNA. It can make proteins independently like the bacteria. So this suggests that mitochondria evolved from ancient bacteria that merged with the early cells. Okay, this is nothing but this is the endosymbiotic theory. Clear? So again a question can come here. Why mitochondria is called a semi-autonomous organelle? So here you can see this is the answer. Okay, now we have discussed about the mitochondria. Now what are the unique features of mitochondria now, which are very specific to mitochondria? They possess their own genetic material. This is a unique feature. No other organelles except chloroplasts consist of their own genetic material. Then they have their own protein synthesis machinery as well, like bacteria. Now they can function somewhat independently of the nucleus. Usually nucleus directs, you can say, all the events that are occurring in the cell, but here you can see mitochondria can produce its own proteins, so it is semi-autonomous. It can work independently. Okay, it can work somewhat independently from the nucleus. It is essential for cellular energy production, so mitochondria produces energy in the form of ATP. Okay, this is the image showing you mitochondria. So you can see here, this is the outer membrane, this is the inner membrane. Mitochondria has its own DNA. This is deoxyribonucleic acid. It is circular DNA. Okay, then you can see it has its ribosomes also. These are the cristae, which are the foldings of the inner membrane. This is the matrix, which is the inner compartment. Matrix also consists of all the enzymes. Okay, inner membrane also consists of the enzymes and the various complexes involved in respiration. This is the intermembrane space, which is the outer compartment. Outer compartment means intermembrane space, means the space between the inner membrane and the outer membrane. Here these are the ATP synthase particles which are present in the inner membrane, especially in the cristae. Okay, now let's move on. Let's talk about the plastids now. Now, what are the general characteristics of plastids? Okay, so plastids are present in plant cell, not animal cells. Okay, and they are present in euglenoids as well. So euglenoids, they are the member of which kingdom? Euglenoids, they are the member of kingdom Protista. Okay, euglenoids is a connecting link between plants and animals. It has both animal-like and plant-like features as well. So euglena example is euglenoids. Example is euglena. So they can photosynthesize in the presence of light, and they can switch to heterotrophic mode of nutrition in the absence of light. They are unicellular eukaryotes. Okay, plastids are large. They are microscope. Okay, they means they can be seen under the microscope. Okay, like for mitochondria, you have to stain. You have to stain the mitochondria, but you can observe chloroplast, which is the plastid, under the microscope. Now, these plastid consist of certain specific pigments which give this plastid different colors. Now, these plastids are double membrane bound structures, like mitochondria consists of two membrane. This plastids also consist of two membrane. It is a double membrane bound structure. So these are the type of plastids you can see. Okay, so these are leucoplast. Okay, these are common plastids: leucoplast, chloroplast, amyloplast, elioplast. Okay, then this is the proplastid, which can develop into chloroplast or leucoplast. Leucoplast is of, you can see, three types: amyloplast, elioplast, and proteoplast. Chloroplast, you can see here it is a two type: chromoplast and the geratoplast. Okay, so these are the type of plastid. We'll discuss about some of the plastids here. Now let's discuss type of plastid based on the type of pigments which are present. First is chloroplast. So chloroplast consists of chlorophyll and carotenoid pigments. So chlorophyll, what is the function of chlorophyll? So chlorophyll is basically involved in photosynthesis. This is the pigment which absorbs the light energy and convert it into chemical energy. Or simply you can say it converts the light energy into food. Okay, so that's why plants and algae, they are photoautotrophs. They can convert light energy into chemical energy with the help of chlorophyll pigment, which is present in the chloroplast. Okay, along with chlorophyll, carotenoids are also present, which are accessory pigments. Clear? Chlorophyll are of two types: chlorophyll and chlorophyll B, and chlorophyll A. This is the primary pigment. Chlorophyll B, this is the accessory pigment. Remember this. Primary pigment is always chlorophyll A only. Okay, what is the primary function of chlorophyll? Photosynthesis. Carotenoids can also assist in photosynthesis. These are also accessory pigments. They also capture light energy and transfer it to the chlorophyll. Okay, now chloroplasts, they are lens shaped or oval structures. What is the size of the chloroplast? 5 to 10 micrometer. Length is 2 to 4 micrometer. Okay, width is 2 to 4 micrometer. Length is 5 to 10 micrometer. Now, the number of chloroplasts varies in different organisms and cell. So chlamydomonas consist of only one chloroplast. Mesophyll cells consist of 20 to 40 chloroplast. So mesophyll cells are present in the leaves. Leaves are the site of photosynthesis. Leaves are green in color. Why they are green in color? Because they consist of chloroplast, and within this chloroplast, chlorophyll pigment is present. Okay, so they reflect the green color. That's why they appear green in color. So chlorophyll provides green color to the leaves. Okay. Here this is the structure of the chloroplast. So chloroplast consists of two layers, outer membrane and an inner membrane. Okay. This is the intermembrane space between the outer and the inner membrane. Okay, this is the internal membrane, you can see. This is the external membrane, also known as the outer membrane. Then this chloroplast consists of thylakoids. This individual one is thylakoid, and stack of thylakoid is known as grana. Okay, this is the granum, you can see here. This granum is a stack of thylakoids. Okay, then it has its own DNA also. Chloroplast DNA is also present here. Then stroma. Stroma is the liquid. It is the fluid present within the chloroplast. Stroma consists of all the enzymes which are required to carry out the biochemical cycles such as Calvin cycle, also known as the C3 cycle. Okay, clear? It consists of ribosomes also, you can see. Now let's move on. Okay, we were talking about the chloroplast. Now let's talk about the chromoplast. So chromoplasts contain fat-soluble carotenoid pigments such as carotene and xanthophyll. Chloroplast, basically they consist of chlorophyll only, okay, some amount of carotenoid, but chromoplast, they consist of fat-soluble carotenoid pigments, carotene and xanthophyll. So these pigments, carotene and xanthophyll, give chromoplast different colors. So it gives yellow color, orange color or red color to the plants. So this chromoplast will give different kind of colors to the plant because this chromoplast consists of pigments like carotene and xanthophyll. Okay, so this is very common in fruits and flowers. So you have seen fruits and flowers, they are of different color because they consist of chromoplast. You can see this is an image showing you chromoplast. Okay, so they have this internal membrane, outer membrane. They consist of the stroma, okay, which is the fluid. Okay, then they have the ribosomes. They have circular DNA. They have this carotenoid crystals, you can see here is present. Okay, carotenoid, there are two types, carotene and xanthophyll. Clear? These are the thylakoid remnants. So this is not chloroplast. Chloroplasts have thylakoids. They have this thylakoid remnants here. So they cannot do photosynthesis, chromoplast, because they consist of carotene and xanthophyll. For carrying out photosynthesis, you require chlorophyll, which is present in the chloroplast. Okay. Now, next is leucoplast. So this is colorless plastid. Now, what are the three subtypes of leucoplast based on the storage function? So based on the material which leucoplast store, they are of three types. Okay, first is the amyloplast. It is a kind of leucoplast only that stores carbohydrates. Example is potato. Then elioplast, they store oils and fats. And then you have aleuroplast, which store proteins. So these are all kinds of leucoplast. You can see here, this is the image showing you aleuroplast and elioplast, leucoplast and amyloplast. Aleuroplast will store proteins, elioplast will store fats and oils. Then you have, this is the leucoplast, which is a colorless plastid, and this is the amyloplast which store carbohydrates. Okay, now let's talk about the chloroplast structure. So we'll be talking about chloroplast. This is the most important. What is the membrane system of the chloroplast? It consists of an outer membrane which is more permeable as compared to the inner membrane. Inner membrane is less permeable. Encloses stroma. Inner membrane encloses the stroma, which is the fluid present within the chloroplast. The stroma consists of all the enzymes. involved in the biochemical reactions. So this is the image showing you sectional view of chloroplast. This is the stroma you can see here. Okay, these are the thylakoids. The stack of thylakoid is known as this granum. In thylakoid, the light reaction occurs because thylakoid consists of all the complexes, okay, and the chlorophyll pigment as well. Okay, this is the stroma lamellae. The stroma lamellae connects the two granum. Remember this. Okay, stroma lamellae, because this is present in stroma. That's why it is known as stroma lamellae. This is connecting to granum. Internal components of the chloroplast thylakoid, these are flattened membranous sacs, and the stacks of the thylakoid, these are known as grana. Okay, thylakoid, they are the site of light reaction. Stroma lamellae, these connect different grana. Lumen, this is a space inside the thylakoid. Okay, so thylakoid consists of a lumen. Lumen is the space inside the thylakoid. Okay, so this is an image showing you thylakoid. This is single thylakoid, and stacks of thylakoid forms the grana. And this stroma thylakoid, you can see a stroma lamellae. This will connect the two granum. Okay, stroma. The stroma, what are the contents of the stroma? The stroma consists of enzymes for carbohydrate and protein synthesis. Okay, it consists of small circular DNA molecules. Like mitochondria also consists of small circular DNA molecules. Chloroplasts also have its own DNA. It consists of 70S ribosomes, which are smaller than the 80S cytoplasmic ribosomes. Cytoplasm consists of 80S ribosomes, but the chloroplast consists of 70S ribosomes, similar to mitochondria. Okay, so you can see here, this also shows that chloroplast was also once a free living bacteria, basically cyanobacteria, which entered into the eukaryotic cell, or which was engulfed by the eukaryotic cell. So origin of chloroplast is also explained by endosymbiotic theory. Okay, now the thylakoids consist of the chlorophyll pigments. Okay, and light reaction occurs in the thylakoid. Okay, what is the distribution of the plastids? So mainly chloroplast. Okay, we talk about the distribution of the chloroplast, mainly found in the mesophyll cells of the leaves. It is essential for photosynthesis and food storage as well, because chloroplast consists of chlorophyll pigment. Chloroplasts are of variable shapes: lens-shaped, oval, spherical, discoid, or ribbon-like. Okay, let's talk about the ribosomes. So ribosomes were first discovered and observed by George Palade in 1953 using electron microscopy. They appear as dense granular particles. Remember, this ribosomes doesn't have any membrane around them. So they are non-membrane bound structures. They don't consist of any membrane around them. They are not enclosed by the membrane. They are composed of both RNA and proteins. So ribosomes are made up of RNA as well as proteins. Okay? They consist of ribosomal RNA. Structural organization, all ribosomes, regardless of their type, have two subunits. So ribosomes have two subunits, larger subunit and smaller subunits. Now these two subunits, they work together during the protein synthesis. So these two subunits, they combine together to form the complete ribosome during the protein synthesis. So these subunits, they can separate and then they can rejoin depending on the need of the cell. So if they require protein synthesis is required, then these two subunits, they combine together to form the complete ribosome, and then they translate the messenger RNA into the proteins. You can see this is the structure of the ribosome. Here you can see this is the complete ribosome, and this ribosome consists of a smaller subunit and a larger subunit. So smaller and larger subunit, they fit together and they form the complete ribosome during the translation process. What are the type of ribosomes? So eukaryotic ribosome, they are of 80S type. So these kind of ribosomes are present in higher organisms such as plants, animals, and fungi. They are larger in size compared to prokaryotic ribosomes. So prokaryotic ribosomes, they are of 70S type. Okay, the 80S ribosome have two subunits, 60S, which is the largest subunit, and 40S, which is a smaller subunit. Together, they make the 80S subunit. Okay, not 100S, 80S ribosomes. Now, prokaryotic ribosomes, they are of 70S type. They are present in bacteria and other primitive organisms. They are smaller in size as compared to eukaryotic ribosomes, made up of two subunits, 50S, which is the largest subunit, 30S, which is the smaller subunit. So together they make 70S ribosome. Okay, what is this S? We were talking about 70S and 80S. What is this S? This is the Swedberg unit. So it is represented by S in ribosome nomenclature. Okay, means ribosome naming. It measures the sedimentation coefficient and it indicates how quickly particles settle in a solution during the centrifugation. Okay, by centrifugation, a pellet and a supernatant is formed. So S denotes how quickly the particles will settle in a solution. It serves as an indirect measure of particle size, molecular density, shape. Okay, so this S is the indirect measure of all of these three: size, molecular density, and the shape. Obviously, if the size will be more, density will be more, then it will settle faster. Okay, now let's summarize this entire lecture. We talked about the mitochondria, plastids, and ribosomes today. So, mitochondria and chloroplast, they are double membrane bound organelle. Outer membrane of the mitochondria, this is smooth. Inner membrane is forms cristae. Okay, mitochondria present all eukaryotic cells. Their size range from 0.5 to 1 micrometer in diameter. Chloroplasts are present in plant cells. They are also double membrane structure which consists of thylakoids. Thylakoids are arranged in stacks which are known as grana. Within the stroma matrix, chloroplasts consist of chlorophyll pigments and they are the site of photosynthesis, and they convert light energy into chemical energy. Chemical energy is in the form of ATP and NADPH. Ribosomes are cellular organelles composed of two subunits. They are made up of ribosomal DNA as well as proteins. They are involved in protein synthesis. Okay, they are not surrounded by any membrane. In eukaryotes, ribosomes can be found free in the cytoplasm or attached to the endoplasmic reticulum. When they are attached to the endoplasmic reticulum, it is known as rough endoplasmic reticulum. In prokaryotes, these ribosomes float freely in the cytoplasm. Now, there are some questions based on these topics. First question is, the inner membrane of mitochondria forms. Options: cristae, cisternae, thylakoids, or matrix. What will be the correct answer? Okay, option number A will be the correct answer. Cristae, not cisternae. Okay, option number A is the correct answer here. Inner membrane of mitochondria form deep fold known as cristae. Okay, so these cristae increase the surface area of the inner membrane, provide space for more respiratory chain complexes, enhance ATP production efficiency, and these cristae, they project into the mitochondrial matrix. So they are towards the matrix. Now the next question, which organelle is self-duplicating? Okay, which organelles can replicate, reproduce? Options: Golgi body, lysosomes, mitochondria, or endoplasmic reticulum. What is the correct answer? Okay, here the correct answer will be option number C, which is mitochondria. So mitochondria are self-duplicating organelles. They contain the old DNA known as the mitochondrial DNA. They can replicate independently. Okay. Now division occurs through binary fission, which is similar to bacteria. Okay. So this supports the endosymbiotic theory of mitochondrial origin as well. This is the structure of the mitochondria. They have their own mitochondrial DNA. Okay, endosymbiotic theory of mitochondrial origin. This we have already discussed. So ancient aerobic bacteria was engulfed by the early eukaryotic cells. Instead of being digested, they form a symbiotic relationship. So both were getting benefited. Now these bacteria evolved into today's mitochondria. Now mitochondria get some of the bacterial traits like DNA and ribosomes. Okay, and this bacteria, this evolved into what is today known as mitochondria, and it generates energy in the form of ATP. Okay, next is chromoplasts are responsible for what are the options? Photosynthesis, food storage, flower and fruit coloration, and protein synthesis. What will be the correct answer? Okay, so let's have a look at the answer. So answer is option number C, flower and fruit coloration. This is absolutely correct. Explanation: chromoplasts are colored plastids. They contain pigments like carotenoids. They give red, orange, yellow colors to the flowers and the fruits, and they help attract pollinators also. Okay. Now they develop from chloroplast or leucoplast. Common in ripening fruits like tomatoes and peppers. This is the image showing you chromoplast. Next question: the site of light reaction in the chloroplast is stroma, grana, matrix, or cristae. Where does the light reaction occur in the chloroplast? So let's have a look at the answer. Answer is option number B, grana. Grana is actually the stack of the thylakoid. So light reaction occurs in the grana of the chloroplast. Grana stacks of the disc-shaped thylakoids. Now, thylakoid consists of chlorophyll pigments and other photosynthetic pigments as well. Photosystem 1 and 2 are located in the thylakoid membranes. Now, grana provide increased surface area for efficient light absorption and the photosynthesis. This is image showing you thylakoid. These are stacks of the you can say stacks of thylakoid known as grana. Now, key notes for you. Now, the oxidative phosphorylation through which the ATP is generated occurs within the inner mitochondrial membrane and involves a series of protein complexes and electron carriers which are present in the inner mitochondrial membrane. Thylakoids, okay, they are present within the chloroplast. Thylakoid membrane consists of all the complexes and the you can say the chlorophyll pigment as well. Thylakoids are organized into stacks known as grana, and these grana are connected by stroma, lamellae. Okay, now the fluid within the chloroplast is known as the stroma. Stroma consists of all the enzymes responsible for carbon dioxide fixation and other metabolic processes as well, such as protein and carbohydrate synthesis. Mitochondria, chloroplasts have their own DNA. Okay, clear? And they contain genes for photosynthesis and gene expression as well. The chloroplast, you can say. Okay, chloroplast is involved in photosynthesis. Prokaryotic ribosomes are 70S, while eukaryotic ribosomes are of 80S type. Okay, you should know about the larger and the smaller subunit present in these kind of ribosomes. Some interesting questions for you. First is compare and contrast the functions of mitochondria and chloroplast, and what are the key differences in their roles within the cell. Next is you have to describe the double membrane structures of mitochondria, And how does the inner membrane foldings on the cristae contribute to the function? Next is, how does the structure of the thylakoid membrane facilitate the capture of the light energy? Next one is, you have to describe the structure of the ribosome, including its subunits and their respective roles in the protein synthesis. Okay, now we have discussed the theory. Now let's discuss some DPP related to these topics. Okay, let's begin with the first question. What is the first question? Which of the following correctly describe the structure of the prokaryotic ribosome? Options are composed of single 70s subunit. Next is made up of two subunits, 40s, 60s. Third is composed of two subunits, 50 and 30s, forming a 70s ribosome. And fourth is made up of three subunits, 30s, 40s, and 50s. What will be the correct answer? The option number C will be the correct answer here. The prokaryotes, they have 70s ribosomes, which are made up of two subunits, 50s, which is the largest subunit, 30s, which is the smaller subunit. Now, next one, what is the function of polyribosomes, also known as polysomes in eukaryotic cells? So, in prokaryotic cells, the prokaryotes, they have polysomes. So, what is the first option? Storage of reserve materials like glycogen. Second is simultaneous translation of the single mRNA by multiple ribosomes. Next is synthesis of ribosomal RNA. Fourth one is formation of gas vacuoles in the photosynthetic bacteria. What will be the correct answer? See, option number B will be the correct answer here. Okay. So polyribosomes means many ribosomes, they attach to the mRNA and they carry out simultaneous translation of single mRNA, okay, to produce multiple copies of that protein. Next is, which of the following is not an example of inclusion body in the prokaryotes? Option are phosphate granules, cyanophacin granules, lysosomes, and glycogen granules. What will be the correct answer? Okay, option number C will be the correct answer here. Lysosome is not an inclusion body. Okay, rest others, they are all inclusion bodies. Okay, lysosomes are known as the suicidal bags. This consists of hydrolytic enzyme. Next one, gas vacuoles are observed in which group of organisms? Okay, all prokaryotes, blue, green, and photosynthetic purple, green bacteria, eukaryotic algae, or fungi. What will be the correct answer? Okay, option number B is the correct answer here. Blue, green, and photosynthetic purple, green bacteria, they consist of the gas vacuoles. They provide buoyancy to these bacteria as they are aquatic. Next one, what is the approximate size of prokaryotic ribosomes? What would be the correct answer? Okay, you have four options A, B, C, D. What is the correct answer? Okay, the correct answer here will be option number B. Clear, 50 nanometer into 20 nanometer. This is the correct answer. Okay, so with this, we have discussed the DPP also. Okay, so we will end the session here. All our answers are correct. Okay, I hope the session was quite knowledgeable for you. We'll meet you in the next session. Thank you very much for listening.